What happened? Oh,
I don't know. I keep listening this electronic. Have you been made show main screen? Show my screen. Okay. I want to
Yeah.
be sure that the
yeah, and I have what the attendee is seeing.
how many you have?
We'll try to
102.
Okay.
Can you hear me yet?
Yes,
Yes.
Can
now
you hear me?
we
Okay.
hear.
Yeah, the last time. Okay, the last time I looked, we had 102 registered.
I think we have 103 now.
Oh, okay, good.
So basically, you're seeing the large screen, right? With the
Now you need to swap displays.
again.
Yeah.
Well, I have. Oh, hold on. Where was that?
Yeah, that's in PowerPoint itself. There's a little the third icon in the upper left corner.
Hold on. The third icon, edit on the upper right corner.
Is swap.
Slideshow. In edit or where?
No, on the on your presenter view,
Yeah.
there's some there up in the upper left corner. The third icon over is swap.
Swap. Where are you? I have insert design transitions animation slideshow. Okay, maybe this is no.
Well, now we're seeing your desktop with connected to GoToWebinar.
Right, but that's I want to swap, so that's what I want to do.
I mean, I saw it on your screen. So
I
now,
want that you see the other one,
oh no.
the actual presentation. How did we do it last time?
Well, underneath in the sharing box, underneath
Okay,
show.
I show you this one now. Now you can see the PowerPoint.
Yeah. So now
Where-
click on your slideshow. Start the slideshow.
Okay.
Okay, now I'm seeing the big slide plus the smaller slide, the next slide up, and then all of the slides at the bottom. So that's your presenter's view. But you
Yeah.
want to go up to the top, right above the big slide. See, it's a swap displays.
Oh yeah. Okay.
Yep, there we go. Now we are seeing it.
Right. Yeah, what I was saying, it's easier for me if it was the other way, but I don't know how to show because this is on the side. What you're seeing now is on the side.
Well, you said the last time you said you wanted to see it in monitor 2.
Whatever, it's fine. Are you seeing me or not?
Yes.
Yeah, we see you and we see your slide.
Okay. So if I switch it back to the other, yeah, I don't know how to do that. Let's leave it like this. Because if I switch displays again, you will see the other one, but I don't know how to do it, such as you see monitor 1.
So up in the sharing box where
Yeah.
there's the play, basically like a play button. Underneath
Yeah.
that, there's show and screen. And I believe you had said you wanted to show monitor 2.
So I put it now in front of you. Can you see it? Sorry, here.
I'm still seeing the large slide.
Yeah, and you don't see the control panel as well?
I don't see your control panel, no.
I put it on top of, in front of the slide.
Yeah, but I'll never see your control panel.
Okay, so what do I do then? You said, what do I do?
So you said you wanted to switch where so you're seeing on one computer, one monitor, you are seeing your
The
presentation,
presentation,
right?
the other, I can see what is coming next.
The full slide, right?
Yeah.
So that means that, so what you're sharing is the full slide, but you want to be sharing the other monitor. So you need to
hold
share.
on. Charlotte is coming, she may help me. Hold on.
Okay.
Yeah, I want to see this in here.
So, Chalet underneath the share
You should
screen.
have different screen option. Try this one.
Now, yes, that's
Then
it. Now click on swap displays again.
Which was here. Show this.
Okay, so now we're seeing just the big slide.
Okay,
And
that's perfect. Thank
Interview
you.
where you want it. Okay.
Yeah, perfect. Because now it's now I don't see any of you, but that's okay.
Yeah. I don't know if Chalet's back on her computer yet.
Yeah,
She
she's
went
asked a
with her.
question.
Just a heads up, the broadcast got started. So right now there's no attendees yet, so we're okay. But the recording started. I hit.
Everyone, and welcome to another webinar sponsored by the NAVBO Education Committee. I am Enar Cuervo from the University of Illinois at Chicago, and I will be moderating today's session. We are pleased to welcome our speaker, Dr. Guillermo Oliver from the Feinberg Cardiovascular Research Institute at Northwestern University. Dr. Oliver will present his research entitled The Lymphatic Vasculature, Developmental Mechanisms and Functional Roles. At this time, I'd also like to welcome Dr. Xiaolei Liu. She's a research assistant professor of medicine, also at Northwestern, working with Dr. Oliver, and she will monitor today's questions. We have something new to announce today, and I'm sure you've received something on your email. Our webinars and our journal clubs, as well as other online activities, are now listed on the NAVBO Events app. It's available exclusively to NAVBO members, so those of you who are not NAVBO members, another good reason to consider becoming members. So be sure to download the app and keep track of NAVBO's online events. You will still be receiving emails and other reminders, but this is an additional way that you can connect with us. During this webinar, the way we're going to handle the questions is in two ways. Throughout the presentation, you can type your questions into the question box on the control panel. These questions will be answered at the end of the presentation. So at the end of the question and answer period, those questions that have been typed off the box, if there is additional time, attendees will be able to ask additional questions real time. So if you still have a question, please raise your hand by clicking on the hand icon on the left side of your control panel. You will be recognized and you can unmute your mic and ask your question. We remind you that the webinar is being recorded and archived on the NAVBO website for future use. Now, even though Guillermo needs no introduction in the lymphatics field, I will like to say a few words. The Oliver Lab focuses on understanding the cellular and molecular mechanisms controlling organ formation in mammals. The research aims to address key aspects related to the development of the visual system and the lymphatic vasculature, both in normal and pathological settings. Almost 20 years ago, the Oliver Lab identified the gene PROX1, a discovery that provided the first specific tools to dissect the process leading to the formation of the lymphatic vasculature. Since then, Dr. Oliver's lab has been at the leading edge of the lymphatics research. His lab's work has been instrumental to demonstrate that most lymphatics are venous-derived and how defective lymphatic vasculature can promote late obesity and associated metabolic disorders. More recently, his lab has identified a role for lymphatics in organ growth and cardiac repair. Guillermo has held leadership roles in major international societies, including the Society for Developmental Biology and at NAVBO, where he has been recently awarded the 2021 Earl Benedict Award. So congratulations, Guillermo. Please go ahead. I would say the floor is yours, but in this case, the screen is yours.
Well, okay, thank you, Enar, very much for your kind words, and thank you, NAVBO, for inviting me for this talk, and thank you for the attendees for joining us for today's presentation. So basically what I we like to do today is to give you an overview of the lymphatic biology field, focusing mostly in the developmental mechanisms and novel functional roles, and a field that has really progressed extensively in the last 5 years or so. And because I imagine the audience is somehow heterogeneous, normally those working in the blood vascular field, they don't need to provide an introduction before they start their talks. However, those of us working in lymphatics still need to provide an introduction because not many people are familiar with the lymphatic vascular network. So I will feel really I accomplished my goal today, but at the end, those of you which again didn't know much about lymphatics feel that is a fascinating and important field with lots of new avenues to follow both in health and disease conditions. So let me start by reminding you that we have two major vascular networks, obviously the blood vascular vessels, and then the lymphatic vascular network, which is the focus of today's talks. And these two networks are, at the naked eye, relatively similar and difficult to distinguish. And partially, the reason why research in lymphatics didn't progress much, I like to think, is because the lack of lymphatic specific markers that allow us to distinguish between these two networks. Just a little bit very briefly about the anatomy of lymphatics. Basically, the lymphatic network is distributed almost everywhere in our body. And just two notes I want to say before I continue. My talk will focus mostly in the mammalian lymphatic vasculature. So although there are major breakthroughs in zebrafish and xenopus and birds, there are certain differences, and my talk will be focusing in mammalian lymphatic vasculature. And because timing constraints, I will not be able to acknowledge a lot of people that contributed during all these years in many aspects. So I apologize ahead of time for that. Anyhow, going back to this slide, basically lymphatics have smaller initial lymphatics, which are, you can see it in this slide, in which basically the endothelial junctions are relatively relaxed, and the McDonald labs at UCSF characterize them and call it bottom-like junctions that basically facilitate the entrance of fluids and macromolecules. And these initial lymphatics eventually connect with larger collecting lymphatic vessels, which are depicted here, in which the endothelial junctions are much tighter and also called zipper-like junctions. And different to initial lymphatics, they are covered by smooth muscle cells and they have lymphatic valves. So basically, you can see here the initial lymphatics then connect to the collecting lymphatics that enter into the lymph nodes with these molecules and fluids are filtered, and eventually they go through efferent collecting lymphatics back to the blood circulation. And then there is another type of specialized lymphatics in the small intestine, which are called lacteals, which are, you can see it here. And these are very specialized, also has bottom-line junctions that facilitate the transport of these lipid-rich particles called chylomicrons that are transported from the lacteals back to the blood circulation, and that's how lipids are absorbed. So regarding the functional roles of lymphatics, this is, I think it's an interesting note just to indicate how slowly the progress in lymphatic research has been in the last 50 years or so. This is a table from a review article I wrote in 2004, which basically summarized what was known about the role of lymphatics in health and disease at that time. And this has been pretty much the same until, I would say, 5 years ago, and I will show you in a minute in which this has changed. But this, even in medical textbooks, this has been the dogma pretty much of what lymphatics do. Basically, lymphatics are considered a sewer system in which they drain lymph fluid from the extracellular spaces. They critical to absorb lipid from the intestinal tract, maintain fluid homeostasis, and obviously the transport of inflammatory cells. In disease, the best known is congenital or acquired defects of the lymphatic vasculature that leads certain diseases, kind of lymphedema. And then obviously they are very well known by their role in the transport of metastatic cells during tumor formation. So this is a slide of what I just told you. What we know are the traditional lymphatic functional roles. We have learned dissect this process better in the last 10, 20 years. And as I told you, it's known that mutations in certain genes or removal of lymph nodes after surgery can lead to lymphedema, meaning swelling, in particular one extremity, as you can see here, in which there is accumulation of fluid and adipose tissue in the area affected. And as I said, also they are very important in the transport of metastatic cells, as you can see here, and also in the regulatory process, immunity and inflammation, as you can see it also here. But probably one of the most exciting aspects of these has been really this flow of new information that happened in the last 5 or so years, in which a lot of sometimes unexpected novel functional roles of lymphatics have been identified. And some of them, and I will describe them a little bit more in detail later on, are summarized here, like the role of lymphatics in obesity and metabolism, also the role of lymphatics likely in inflammatory bowel disease, particularly in Crohn's disease, also a specialized lymphatic in the eye called the Schlemm canal, that when defective leads to glaucoma, really emerging role of lymphatics in cardiovascular diseases, and also relatively novel and unexpected, the role of a specialized lymphatic on top of the meninges called the meningeal lymphatics. And this has been now linked to a variety of neurological disorders, including Parkinson and Alzheimer's. And then I included there to be determined. I'm pretty certain that in the next years or so, we will keep identifying novel and unexpected functional roles. And I will describe towards the end the latest one my lab identified, which is in organ growth. So all this to tell you that this is a really dynamic field that keeps progressing and progressing and is getting more and more exciting. So regarding historical perspective, and I think sometimes it's important for, particularly for those young students and postdocs to get a sense of how history happens in certain fields. And I always insist that it's so important to go back. We all rely on review articles sometimes, but going back to the original literature is very important, and we can learn a lot. And evidence suggests that the first description of what we think was indeed a lymphatic was by Aristotle in the 300s before BC. But the real clear definition of a lymphatic was with Aselli in the 1600s, in which he described something that he called it white veins. And there has been, as you can see in this graph, a number of events that happened, but clearly until the 1900s, 1902, I would say nothing major happened regarding the description of the lymphatic vasculature by itself. And then at that time, a remarkable woman, and I will tell you more in a minute, Florence Sabin, proposed the first model to describe how lymphatic development occurs in the mammalian embryo. In 1910, there was an alternative model proposed by Huntington and McClure that suggested that, as I said, an alternative model. And then we jump almost 100 years. Again, that not much happened regarding developmental aspects or innovation in lymphatics. And as I told you, partially probably because of the lack of specific markers. But what was basically a Littlus lab, Kari Littlus lab in Finland, identified vascular receptor 3 as one specific lymphatic marker. It became a valuable tool at that time. And then in 1999, my lab identified the transcription factor PROX1 as the first specific lymphatic marker and also the first mouse model totally devoid of lymphatic endothelial cells. That's more or less the timeline in which we are. So you can see there are big gaps in all of these. Anyhow, so how we started into this. So basically, for reasons I don't have time to go around, was when we identified this gene PROX1. And this was done at that time by Jeff Weigel in my lab, a postdoc in my lab, that he showed basically that this was, you can see it here in green, a specific marker for lymphatic endothelial cells. And you can see in purple the blood network intermingled with the lymphatics. And that was per se already quite an important achievement, because now we have a valuable marker. But at the same time, what was even more interesting was that he found that embryos at 14.5, that's when they died, the PROX nulls, show extensive dermal edema, indicating some type of major lymphatic problem. But basically what he demonstrated, that this embryo was totally lacking lymphatic endothelial cells. However, the blood vasculature was perfectly normal. So this was the first description on an animal model that completely lacked lymphatic endothelial cells. But again, that's why I show you the historical perspective. This was quite challenging at that time, because we didn't know much about lymphatics. There were no markers, so it was very difficult to characterize this phenotype in further detail. So what we did extensively with Jeff was to go back to the old literature, the one I just was telling you. And in particular, I became absolutely obsessed with this remarkable scientist, Florence Sabin, because as I told you, in 1902, she has been doing this remarkable ink injection, in particular in pig embryos, and these are her original drawings, which are outstanding. And she provided a detailed description of how the lymphatic vascular network is formed in the mammalian embryo, and she proposed that they have a venous origin. And I really, I strongly suggest you guys read about her. But I became so obsessed, and those that know me and those that came through my lab knows that when you join my lab, I give you this book that is a small book that basically summarizes the major work of Florence Sabin regarding the characterization of lymphatics. I really encourage everybody to read it. It's a joy and a jewel to read it. But something I want to stress, if we are all aware today how challenging it is for women to do science many times. Imagine, unfortunately, extremely challenging. Imagine in the 1900s, how difficult it was for this lady to do what she was doing. And this scientist, this remarkable scientist, I just summarize here some of her accomplishments. She was the first woman to be elected president of the American Association of Anatomists. She was the first woman elected to membership to the National Academy of Science. I was the first woman appointed to the faculty at the Johns Hopkins University of School of Medicine. And I want you all to read this statement that she wrote in 1929. I hope my studies may be an encouragement to other women, especially to young women, to devote their lives to the larger interests of the mind. It matters little whether men or women have the more brains. All we women need to do to exert our proper influence is just to use all the brains we have. I really hope that same way that Oprah Winfrey supported a film about the HeLa cell, Henrietta Lacks, one day somebody will make a movie about Florence Sabin. I think it will be a model that many people will follow, and it was a fascinating, tremendous scientist and an amazing woman. Anyhow, as I told you also, in 1910, there was another model proposed by Huntington and McClure that basically challenged what Sabin said, and they proposed basically that lymph sacs arise in the mesenchyme independent of the veins, and then they establish venous connection. And as you will see in a minute, this debate is still going on 100 years later. But just to get a sense also, sometimes we feel that our colleagues are a little bit mean to us or things like that. Read here what Huntington and McClure said about Florence Sabin's papers. Considering our present knowledge of their development, it is it is evident from the very nature of the case that the injection method, as used by Dr. Sabin, has not yet given us the slightest clue as to the actual manner in which the unlages of the lymph sacs are derived from the veins, nor, in fact, any evidence at all that they even possess a venous origin. So just that to put you the framework in the context in which Florence Sabin was working with and how strong this debate was. Anyhow, going back to our findings in 1999, basically that was the type of science of literature Jeff and I we were following to try to really characterize why these embryos lack lymphatics or how lymphatics are formed. So this was the first indication supporting Sabin's statement when basically we determined that the process of lymphatic formations starts here in the mouse embryo. This is a transverse section at the level of the anterior cardinal vein. And what you see in red is beautiful Prox expression in one side of the cardinal vein and not in the other, which already poses a fascinating question. We still are not sure why it happened, why it's on one side. But this is what we today call lymphatic progenitors. This is the first step leading to the formation of the entire vertebral lymphatic vasculature, the presence of Prox-expressing cells in the venous endothelium, which in the mouse starts around E10. So obviously, as you can imagine, this poses a lot of questions for us of why is this happening, or how does it happen? So following on Sabin's steps, we propose that the step of lymphatic formation in mammals follow 4 steps: the specification of progenitors in the cardinal vein, as you can see here, then the budding in a very polarized and specific manner of these progenitors to form 7 different lymph sacs along the anteroposterior axis that eventually will form the entire network. So then again, just to give you a perspective of how things were at that time, this is again from Jeff's paper. Based on the knowledge at that time, we are developmental biologists by training. So we use the concept of developmental biology, trying to summarize or put a scheme of how lymphatic endothelial cells originate. So basically in developmental biology, the concept of cell differentiation starts by a cell acquiring competence. So basically something happens, so this cell will be ready to start to become whatever type of cell it is, in this case, a lymphatic cells. Therefore, that cell become biased, and eventually by adding additional markers become specified and in the past to be fully differentiated. What we suggested in 2002, based on the few markers we have, David Jackson at that time in the UK showed that LIVE1 is also expressed the hyaluron receptor in the cardinal vein. So we suggested that that's the initial competence of the vein to become lymphatic. Then Prox starts expressing also VEGFR3 in one side, Prox in one side of the vein. We saw this is the initial progenitors divide, and then the cells start to bud off. So all what we know that was VEGFR3, Prox, and LIVE1. 2 years later, we make this a little bit more sophisticated, and this schema we said that around 9 to 10.5, Prox expression make the presence of lymphatic progenitors. And in Carey's lab in 2003, they show basically that VEGF-C, the ligand for VEGFR3, was indispensable for the formation of lymphatics. When you delete VEGF-C, the lymphatic endothelial cells do not move away from the vein. They remain all stuck in the vein. Lymphatic cells form, but they don't move away. So we added this new marker here. As you can see, we know in the mesenchyme there is VEGF-C, which is required for that body. This was the model we have in 2004, more or less, has been improved, but this is more or less where we stand. And now it is, as I said, it's a little bit more sophisticated model. We start in the vein again, in which we know based on work from my lab and also from Francois' lab in Australia that SOX18 is also important to induce Prox expression, although they are not polarized in the vein. And then later on, through VEGF-C, these cells start budding out. They start expressing additional markers, including podoplanin, and then eventually start forming the sacs and the entire network. So that's what we think about lymphatic development. And again, many other markers have been added by many labs around the world. So When Satish joined my lab at that time, what he decided, based on what I just told you about this debate of venous origin, mesenchymal origin, was to use lineage tracing to really define what happens regarding the origin of lymphatics. And he was able to demonstrate, and I will stress this very much, that what we propose that is in the mammalian embryo, at least we think that, and we stand for that, most, if not all, mammalian lymphatic endothelial cells are venous derived. I know some people will disagree with this statement, but because in the last few years, there has been proposal that other sources also produce lymphatic endothelial cells. For example, for the skin, it has been proposed that a certain percentage, a small percentage, may have other origins, although also Francois' lab has, together with Natasha Harvey, has shown that they seem to adopt from a capillary bed, suggesting that those others also could be from venous derived. There has been proposed that maybe hematopoietic origin for subpopulation of lymphatics in the heart and in the intestine. And in the case of the heart, well, that's probably the most convincing evidence has been coming from Miguel Torres' lab in Spain, showing that in the ventral heart, lymphatics from the ventral heart, 50% of them originate from the second heart field. That they are also dependent on PROX expression. And that is very conclusive at the functional level when they did PROX1 using a second heart field Cree line. Basically, there are no ventral lymphatics. So all what I'm saying, and for the intestine, again, as is a tissue that is constantly regenerated, maybe there are other sources. But just a few years ago, also Oliver Stone at that time in Didier Steiner's lab showed nicely using PAX3 Cree that basically the paraxial mesoderm, basically the lateral plane mesoderm that we know is the source for endothelium, basically give rise, as suspected, to the venous lymphatic endothelial cell, from which PROX eventually will give rise to most of the lymphatic network. And they also suggested that basically also this form a dermal capillary bed, and some of those lymphatics eventually originate directly from that. So my statement on all of this is that we still stand that we believe that most embryonic lymphatic endothelial cells are venous derived. Could be other sources. The contribution may be minimal, and it needs to be proved whether they are relevant at the functional level when the proper Cree lines are available. It could be possible, however, that in adult stages or during injury conditions, in those cases, yes, other sources may be important, in particular, as I said, in the intestinal bed. Anyhow, going back to the original statement, this is what we knew at that time, basically, that lymphatic, as I told you, the formation of the lymphatic network starts here in the cardinal vein with the polarized expression of PROX1. This for us was a fascinating, it's still a fascinating question. Why is on one side and not in both? So possibility is obvious that something induces PROX on one side, or that the populations on each side are different. And the other step that we are fascinated is this: how is this budding occurring? So let's go one step at a time. So to try to address the issue about why is PROX on one side, or whether there are differences in the cardinal vein, Noelia Escobedo in my lab years ago, and this is unpublished data, because we couldn't sort cells to really define positive versus negative sides, so it was not enough. So she did the painstaking tool of laser capture microdissection using the PROX tomato line. And basically what she captured was these four populations: the PROX negative side, the PROX positive side, progenitors, and the budding cells outside of the vein. And then she also captured mesenchyme, because we thought that as she laser captured this, she will capture in the mesenchyme underneath. So this will be basically to remove the background. So these were the four types of cells. She also used four different genotypes. The wild type, the PROX heterozygous, that we know there is a phenotype, and we know PROX is haploinsufficient and dosage dependent, and the nulls. And this is the best model because Now we can really compare a cardinal vein in which prox was never present and really determine whether there are differences between both sides. And we did all these comparisons. I don't have time to go in detail. I just want to summarize the main results. The main results are the major difference as expected are between the cells outside here, those that are budding up, and the cells inside the cells, the progenitors. And this is because as the cells bud up, they start acquire additional markers. Another important thing is that basically the role of prox is to be a repressor. What becoming a leg progenitor means, a lymphatic endothelial cell progenitor means, is to basically downregulate the the venous endothelial cell fate, and that side basically downregulated most of those genes and expressing prox. That's why it's sometimes very difficult to define what a lymphatic progenitor is, because a progenitor is basically a cell that represses the blood endothelial cell program, expresses prox, LIFE1 and VEGFR3. That's what a progenitor is, but mostly the role of prox is to downregulate that fate. Basically, the other important thing is that there are no obvious difference between both sides of the vein before prox expression, so that's not the reason why prox is polarized. And the other interesting result was that somehow, we don't know still how, prox expression on this side influenced the negative side. Anyhow, that's where we are, and we are trying to eventually continue in this area of research. But what I told you, that prox is mostly a repressor, this somehow explained this finding we did many years ago, that what we show is that prox expression needs to be constantly maintained in lymphatics in embryos, postnatal, and in adults. And as soon as you remove prox, cells basically become blood endothelial cells. So prox is constantly required to maintain the lymphatic endothelial cell fate, which is plastic. And the reason why is because you have this underneath venous endothelial cell fate, which prox is repressing. You shut it off and they come back. So now I told you this is still a work in progress of trying to understand how these progenitors are specified, what happens, why is on one side versus the other. The other fascinating step is this of the body of these lymphatic progenitors. This is always in a very precise manner. But also, I want you to think for a minute, and this is something that has always puzzled me. And you can see here a scanning EM image of the cardinal vein. We know that lymphatic progenitors in the vein barely proliferate, and you can see it here in red. And Ying Yang, when it was in my lab, she nicely showed that basically this process of cells coming out from the vein is an active budding mechanism in which cells have really tight adhesion junctions, and as they leave the cells, the vein, as you can see here, they immediately start expressing other markers, but are also tightly interlinked by expression of E cadherin. So it's an active budding process, but the key question is, if these cells will keep budding, you will keep forming progenitors and budding constantly, eventually the vein will disintegrate. So how is this process regulated? This is also another major question we were very curious in the lab. And again, this is mostly what I just told you before. And basically, we know this process of budding, as I said before, is regulated by VEGF-C, which is expressed in the mesenchyme. And as I said, Kari's lab showed that very nicely in the mutant VEGF-C, and Shingen in my lab also did in situ hybridization. And what you can see at the bottom is the double in situ in blue for VEGF-C and in dark brown prox expression. You can see the path of these budding cells really follow the higher levels of VEGF-C expression. But this is even more complicated because VEGF-C to be active need to be proteolytically processed, as shown by Alitalo, Mark Kahn's lab. You have these processes, Adams and CCB1, that needs to really cleave and activate VEGF-C. So this is another fascinating question. How is this precise budding regulated and controlled? Anyhow, but just to figure out why the vein doesn't basically implode and disintegrate, how do we control the number of progenitors? Again, so this basically show in the lab, and all this is published, basically first that prox dosage really control the number of lymphatic progenitors in the vein, and also the formation of the lymphovenous valve that he nicely characterized. He showed that a subpopulation of those prox-expressing cells don't leave the vein and will give rise to the lymphovenous valve, and he continued that in his own lab. But this is, for what I just was telling you, probably the most important aspect. He showed that there is an active prox-VEGF3 feedback loop, which is required to maintain the identity and the number of lymphatic endothelial cell progenitors. Basically, what he was able to show that VEGFR3 is a dosage-dependent target of prox. He found prox binding sites in VEGFR3. The opposite, that VEGFR3 regulates also prox expression in progenitors. So what we proposed, or what he proposed at the time, is that between 9 and 13.5 in the mouse embryo, there is this active prox-VEGFR3 feedback loop that somehow regulates the formation of progenitors and the budding of progenitors. Somehow, and we didn't know how, after 13.5, this is shut off. We know that there are not more progenitors forming, there is no more budding. What you have is continuous proliferation of the lymphatic endothelial cells that already exit the vein. But how is this regulated? It was something that was really puzzling. And I will give you just a hint. And this is work from Wenshu Ma, a very talented postdoc in my lab. And this hopefully will be published soon. And some hints of this was given by Peter Carmenet's lab years ago, suggesting that metabolism may have something to do with lymphatics. But basically what Wenshu has shown now, that basically is the microenvironment of these migrating lymphatic progenitors and regulating mitochondrial respiration that is absolutely critical to control this feedback loop and to determine when to shut it off, and basically when there is no more specification and no more migration of lymphatic progenitors. As I said, hopefully this will be impressed very soon. So summarizing, this is where we are, a more sophisticated model, where I just told you what happened early on, and then what happened during migration, these autoregulatory loops. And obviously, all this work has allowed, from many labs around the world, has allowed to identify a number of key players involved in different aspects of lymphatic formation. And many of those have been found to be mutated in different forms of lymphatic diseases and lymphatic malformation. This is a small list indicated here. And now, for the rest of my talk, I just want to focus on what I said at the beginning, which is this plethora of novel functional roles identified for the lymphatics. And I think this is a very exciting field, and it will continue to grow in years to come. And I would like to think that we somehow contributed at the beginning of this, and this was work done by Natasha Harvey, when I was a postdoc in my lab, when she characterized it. What we noticed is that the prox heterozygous mice have major lymphatic defect, but besides, they became extremely fat with age. We didn't know exactly why. It took Natasha a long time to figure out, but bottom line, she found that the lymphatics in these animals were defective, were leaky, as you can see here, chyle leakage. And basically, she showed that these lymphatic have major holes, and that's where they are leaky. And all the chylomicrons, basically the lipid-rich particles that should be inside the intestine, leaks out. She demonstrated that in vitro, this chyle is adipogenic. Bottom line, what we proposed it at that time, and this was followed up later on by Noelia Escobedo in my lab, was that there is a novel cause of obesity, which if your lymphatics are leaky, you have a subtle leakage of chyle, you become obese. I know there has been, many colleagues didn't really accept this idea. There was not a lot about obesity and lymphatics, although there are very clear evidence that there is a crosstalk between these two, because in individuals with edema, it's known that the area around edema accumulates fat, suggesting some type of crosstalk. So, but the question was, maybe this is only in prox mice that have many problems. Does it happen in humans, etc.? So this is something that it was a valid question and we wanted to address, but obviously this was not an easy question to address. So what we decided to do, Heijin, Jill, and Wenshu in my lab, was to see if we can identify biomarkers that will give us a hint that lymphatics are defective, because the problem is to identify lymphatic defects. Until now, most of the assays are really invasive, like lymphangiography, and you cannot do that in any individual that you want to test. So the idea was to see if there were biomarkers that will tell us that in this individual, symptomatic or symptomatic defects of lymphatics are present. And we decided to use exosomes. The idea was that maybe these biomarkers were very diluted otherwise, and then in exosomes may be concentrated. We used blood exosomes, and we started using mice. We use wild types, which are normal, procs, prior and after the obesity, and OB mice, which are the leptin receptor mutant, which are extremely fat, but they have normal lymphatics. We did exosome purification in collaboration with Hector Peinado in Madrid, an expert in exosomes, and proteomics analysis, and we find a number of markers that were somehow specific for the OB mice. And then we started, okay, what was the issue in humans? We collaborated with Stan Roxon at Stanford University, which has samples of patients, and I know we are very aware this is a small sample number. It's not easy to collect samples and it's expensive. Somehow we have, as you can see here, normal subjects, patients with lymphedema, vascular disease, and some with lipedema. And lean and obese were included as part of that study, and the same analysis of exosome was done. And the really rewarding finding was that we found that platelet factor 4 was consistently elevated in mice and human in all individuals with defective lymphatics, but was perfectly low, normally low in all normal controls. So, and also interesting was also increase in lipedema patients, suggesting that lipedema may have a lymphedema underlying defect. Again, I agree that the numbers are still low, but this is suggested that we have a biomarker that will allow us to identify those patients. And basically, this is the summary of that. And one thing we include in that paper is that basically many of these patients, Stan has been doing a table about that with different lymphatic diseases have a number of associated comorbidities, as you can see it listed here. And the fact that in the last 5 years, a number of diseases had been associated to lymphatic function suggests that maybe patients with lymphatic defects, you should expect to see associated comorbidities. What we would like to do in the future is take advantage of this biomarker to basically use a large number of individuals, in this case, cases in which there are patients with symptomatic lymphatic defects, but also others in which, like our prox mice, they don't have obvious lymphatic defects. They are just obese, like in our mice. And in those, basically take blood samples, compare the levels of PF4, and basically see what type of associated comorbidities are present in those individuals to try to see if we can really link in a definite manner a variety of lymphatic disorders with malfunction, symptomatic or asymptomatic of the lymphatics, including obesity. And that's where we are in this aspect. And now the last part of my talk, I want to tell you about the latest functional role of lymphatics in organ growth. And this is work that was done mostly in my lab by Xiaolei Liu, extremely talented postdoc. I just want to tell you that she's in the job market for starting her initial assistant professor position. She is a remarkable scientist and a terrific colleague to have. If you are interested, contact her directly. And we were helped also by Wenshu and Michael in our lab and a lot of people I will cite in a minute. So, but basically the idea was how the lymphatic development occurs. Those of you who are not familiar, already Paul Riley has described it before, and Xiaolei also did it here with LIVE1, a lymphatic marker. We know lymphatic formation starts in the mouse embryo around E14.5 on the dorsal side, and by P0 mostly covered the entire surface of the heart. And as I said before, lymphatics now have been linked to a variety of lymphatic defects, including atherosclerosis or myocardial infarction. And for example, it was reported that basically by Paul Wrighty's lab, that they respond at the increasing numbers after cardiac injury, that stimulates the resolution of inflammation after MI. And also, if you increase lymphangiogenics, for example, using VEGF-C, you improve cardiac function after MI as well. So there were a lot of evidence suggesting a crosstalk between lymphatics and cardiac function. So what Shirley decided to do was to take advantage of our prox conditional mice and delete it at the moment in which lymphatics start to invade the heart, meaning tamoxifen injection around E13 and 14.5 using V-cadherin cream, and then analyze at E17.5 the null conditional mouse. And this was already a surprising result. As you can see very nicely in the heart, you can see in controls nicely in lymphatics in the surface of the heart. However, in the nulls, you can see there are no lymphatics whatsoever, but the surprising result was that the heart was 1/3 smaller. And that was really surprising result, totally unexpected. And 1/3 smaller is a lot. You will die with that size of a heart. Obviously, the argument was prox does a lot of things, is expressed in a variety of tissues, maybe it's prox specific. So we collaborated with Sultan Jakri's lab and Laszlo in Hungary, that they provide us with the VEGFR3 mutant embryos, that the KDKD that has a mutation in the tyrosine kinase domain, and basically VEGFR3 is inactive. And as you can see here, basically these animals also, they don't have lymphatics. And nicely, also we found that in this model, the heart was as well 1/3 smaller, supporting the results we find in proximal mice. So why are the hearts smaller? Basically, the anatomy of the heart is perfectly normal. It's just smaller. What Shirley was able to show using different proliferation markers, that clearly proliferation of cardiomyocytes was very reduced in all the mutant embryos. Also, she showed, as you can see here with caspase-3, that apoptosis was increased in this mutant embryo. So that somehow explains why the heart is smaller. So all this data suggested that in the same way that we showed for blood endothelial cells that they produce angiocrine signals, maybe lymphatic endothelial cells produce some paracrine signal, lymphoangiocrine signals that talks to cardiomyocytes, and this is required for cardiomyocytes growth and cardiac homeostasis. To test this possibility, she grew lymphatic endothelial cells, she cultured, she took the conditioned media, and then she added the conditioned media to either iPS-derived cardiomyocytes provided by our collaborator Paul Burridge here in Northwestern, or primary cardiomyocyte. It was very nice. She saw that basically this induced the activity of pro-survival pathways like phospho-ERK and phospho-AKT, as you can see here for iPS or here for primary cardiomyocytes. So, and she confirmed that indeed what this conditioned media does is promote proliferation, you can see it here by Ki67, or reduce cell death, you can see it here as well by caspase. So obviously the next step was, what is this signal that is promoting this activity? We did mass spec and proteomics and bioinformatics, and a number of candidates came out, and we are working in some of those, but the one I will focus on, and we focused for this paper, was reeling. And this was basically initially a somehow surprising candidate, because mostly reeling was known for the roles in the CNS. Reeling mutant mice are ataxic and they die soon after birth. So, and it's expressed strongly in the CNS. So, but Tahya Makinen's lab had published in 2012 that indeed reeling was expressed in lymphatics. Therefore, Shirley decided to follow on that, and indeed she showed that reeling was nicely expressed together with LIVE1 in the lymphatics in the heart as well. And but obviously, following this, the key experiment was now, okay, if that's true, what happened in reeling mutant hearts? Are they smaller? And this was a major tour de force, to be honest, because of the pandemic, a lot of issues. And we really want to thank our collaborators, UT Southwestern, Joachim Hertz, and Undine Cleaver and Cheryl Good for their tremendous help in this part of the project. But basically, we were able to, Shirley was able to demonstrate that really conditional null embryos generated with PROX3 are basically deleted in lymphatic endothelial cells. The heart was also smaller because of the same reduced proliferation and increased apoptosis. That was a major support for the idea and the findings that Shirley has generated so far. So how is really working? Basically, in neurons has been shown that mostly act through these receptors, ApoE or VLDLR, but also has been suggested it may work through the integrin beta-1 receptor. And this was interesting because Deepa Srivastava's lab in California showed years ago that mutant hearts in integrin beta-1, basically cardiomyocyte, the heart is also smaller. That was really suggestive that this could be the pathway. And indeed, Shirley was able to generate the double heterozygous mutants and showed that in this case, the same. The heart is smaller, only the heart, but in other organs, the size of the organs is absolutely normal. So that what this allows us to conclude that basically lymphatic endothelial cells produce lymphoendocrine signals that act on cardiomyocyte. That signal is renin that acts to the beta integrin receptor and promotes proliferation and growth. So to summarize this part, we can say that cardiolymphatics are required for embryonic heart growth, and we think probably for other organs as well. Renin is the secreted factor that regulates cardiomyocyte proliferation and survival through the beta integrin 1 signaling in cardiomyocytes, and renin conditional nulls also phenocopy the heart growth defects observed in lymphatic deficient mutant embryos. So all this is great. So now I told you that lymphatics have been shown to play major roles in cardiac diseases. So the idea was to see what happened during regeneration and repair. Does renin do something? And basically, just to summarize what is known about cardiac regeneration, we know zebrafish regenerates quite well. In mammals, in mice in particular, we know the neonatal heart up to P7 can regenerate, but after that, regeneration capacity is lost in adult mice. And this was, the neonatal case was interesting because when Shirley checked the expression of renin, this was very suggestive because, as I told you before, it's nicely expressed at E17. You can see here together with PROX1 in the heart. It started to get decreased a little bit in the lymphatics around P2, but you still can see it. Same is true at P7, decreases even more, but you still can detect it, but it's absolutely gone by P14. And this matches quite well the capacity of regeneration of the neonatal heart, and that was quite suggestive. The other interesting thing that renin is reinduced if you do a myocardial infarction, renin is reinduced in the injury area very strongly, as you can see it here versus the non-injured area. So all this promote the idea, okay, let's see what happened during neonatal MI and during adult MI. And to do the neonatal MI, we have help from a remarkable student from Spain, Esther, that from Miguel Torres' lab, that she was very skillful to do this very difficult technique of 2MI in P2 PAPs, which is very tricky. And basically what we were able to demonstrate that we did this in really null PAPs, of course, that basically cardiac function is not very good in these really null PAPs after MI. And also basically the proliferation was reduced, apoptosis was increased, and the fibrotic area was also much more increased in this animal. That's why it's explained they have reduced cardiac function in these mice. Then Shirley decided to see what happened in adults. And as I told you, really is highly expressed, as you can see here, in the newly formed lymphatics in the infarcted heart. So to test whether renin has a role in the newly formed lymphatics, she used an approach that was taught to her by Pilar Ruiz Lozano of RegeneCore in California, which is the use of collagen patches to apply a recombinant protein in the heart. In this case, Shirley decided obviously apply renin in those patches, and basically then she did MI and sacrificed the animals at 42 days afterwards, and she measured cardiac function every week. And this was very encouraging because obviously cardiac function was much more improved in those animals, which you can see here in orange, that have the reeling recombinant protein versus those with the control. And basically, the reason for the recovery, we think, is because cardiomyocyte cell death was clearly reduced, but also, and consequently, the size of the fibrotic area was much more reduced, as you can see here. So in summary, basically, I think what we demonstrated is really is required for neonatal cardiac regeneration after MI by improving cardiomyocyte proliferation and survival, and also improves cardiac repair and function by reducing cardiomyocyte cell death after MI. So one thing we think, everybody trying to improve cardiac function after MI is trying to use stem cells or improve proliferation. One thing that we think this also proves that reducing cardiomyocyte cell death is also a very important aspect to consider into this. So bottom line, I just show you another functional role of lymphatics, totally unexpected. And I'm pretty sure in years to come, we will keep finding more and more. And finally, to acknowledge all our people that came through my lab through all these years and all our terrific collaborators here in Northwestern, UT Southwestern, Stanford. I think I mentioned all of them through my talk, the funding sources in all these years. And basically, I'm open to questions, and I encourage to attend the next seminar by Karen Hershey in April 1st. Thank you very much.
Thank you so much, Guillermo. That was a great overview of the lymphatics and a great addition of your work. I think it had both stimulating ways, like a great overview for our NAVBO Education Committee supported webinars, and really exciting science. I have a question for you, if you don't mind. You've seen the effects of reeling from the lymphatics in the cardiac muscle. Have you seen whether the skeletal muscle is also affected by lymphatic senior mutants, or is it specific to cardiac muscle? Because all these muscle dystrophy diseases could certainly benefit.
Sure. To be honest, I don't think we have looked carefully into that. Also, as far as I remember, I think in the beta-1 integrin mutant mice from DPAC, they didn't see difference in the skeletal muscle. So it seems to be very specific for cardiomyocyte. I think the beta-1 integrin receptor is only expressed in cardiomyocyte and not in skeletal muscle.
Okay, really interesting work. At this point, I'm going to yield to Xiaolei, who we're lucky to have the first author, Xiaolei, with us, who's going to moderate the question. So Xiaolei, whenever you want.
Hi. Yeah, I saw several questions through the chat. And the first question is from Lysandra Villa Alice. And she's asking, does overexpression PROX1 in blood vessels, for example, artery, vein, or capillary, induce the lymphatic effect?
Yeah, it's a very interesting question, and one that we have been dealing for a long time, and still I feel inconclusive, to be honest with you. One thing I can say for sure, if you put it in arterial endothelial cells, nothing happens. Absolutely nothing. So obviously the arterial fate is not has no competence to become a lymphatic. We also have put it in cardinal vein, all over the cardinal vein. And clearly we see more progenitors on the vein when we do that. However, I'm not so sure we see more lymphatics in the negative side of the vein. So mice, but this is complicated again, because the issue is we don't have, besides PROC, we have nothing else to define what a progenitor is. Therefore, when we overexpress PROCs on the vein, how do we know if there are more progenitors? So what I can say, we see more PROCs on the negative side of the vein. So, and we are doing additional experiments on that, and we will probably do it again by single cell RNA-seq or laser capture to see if we change the profiling of the negative side. But at the moment, I will feel that it's rather inconclusive whether PROX is sufficient to promote lymphatic fate in other venous beds.
Okay, another question is about the reeling. Does reeling work as an endocrine factor in other organs? This question from Harish Palati Janahai. I'm sorry if I pronounce your name wrong.
Well, there's a another interesting question. Well, if you look at Cholet's paper, what in some of the figures we also focus. So she also focused on the liver and lung, I think, and sometimes in the kidney as well. And in some occasions, we saw some difference in size, although there were no significance. So you can take it two ways: either that they have no role, or that one is that we need really to analyze many more samples of those to reach a conclusion. Or the other could be that 17.5 or the induction at 14 and 13 is not appropriate for those organs. I think it all depends at what age each organ is expanded. So maybe to see an effect in other organs, we need to really remove in other stages. And some of the things are the kind of things we are doing at the moment. But bottom line, I cannot give you a conclusive, but I would be surprised, A, if really is specific only for the heart, and B, if there are no other lymphoendocrine factors which are required for other organs.
And a question from Mark Hunt. Great talk, Guillermo. Do hearts with endothelial loss of PROX1 or mutant VEGFR3 have abnormal coronary vessels?
They have abnormal coronary vessels? That was the question?
Yes.
No, well, you know Cholet as well as me. As far as we know, they are perfectly normal. Mark.
Yeah, for the PROX1 mutant, we did a conditional analysis.
Yeah, this in the conditional notes, Mark. Just the standard null, as you know, they die at 14.5.
I have two more questions from the same person, Amanda Palazzo. And the first question is, have you considering look into the presence of Weibull plate bodies in lymphatic endothelial cell, similar to that of endothelial cells, leading to the increase in PF4 associated with lymphatic damage in patients?
No, we haven't. No. It's a valid point, but we haven't.
How about utilization of intravital microscopy to study the lymphatic system? And do you have any advice on how to do it with such small vessels in the lymphatics?
Well, yeah, that's a challenge working with mouse embryos. I mean, if you want to do it in embryo, it's very, very, very difficult, in fact, to do intravital imaging. And there are a few colleagues that, particularly those working in physiology, that do a little bit of that, but is very, very difficult and very challenging. And also, this brings an interesting question I always have been wondering, which is, we know that if, as I showed, that lack of lymphatics leads to edema, indicating that lymphatics are doing something. But at what point are lymphatics function, not lymphoendocrine factors, but lymphatic transport and lymphatic function becomes relevant in the embryo, is something we still not totally clear. But unfortunately, I don't have any good advice to give you on how to do it. I remember when we published our initial paper in 1999, one of the reviewers asked us to do that, to really visualize what was happening with the lymphatics. And we did it through one collaborator who was extremely skillful and did ink injections through the heart. But again, it was very challenging. So for that, you will probably, if you want to do that, go to larger animals. That's my suggestion.
Oh, I just saw another two questions come out. The one is from Xiao Gu. Collecting lymphatic endothelial cells express reelin that also help recruit smooth muscle cells. When treating with reelin patch, does that affect smooth muscle cells on collecting lymphatic vessels?
Oh, Cholet, have you looked at that?
I haven't. We just saw like overall the morphology of lymphatic vessel is comparable, but we haven't checked for the maturation of the bigger vessels. Good question though. Another question from Joseph Radkowski. Great talk. There are several published studies using VEGF-C to expand lymphatic post-cardiac injury. Would you suppose that improved the improvement seen in these models would be reelin driven as opposed to lymphatic fluid or immune roles?
Well, great, absolutely appropriate question. And that's something Charley is really trying to address, because obviously, based on our data, one thing you can argue is when you promote more lymphatics through VEGF-C or any other pro-lymphangiogenic factor, what you do is you increase the amount of reelin, and therefore you see what we see. So Charley is using a number of approaches to really conclusively address that question, whether the effect of VEGF-C is exclusively mediated through reelin or is not. And hopefully soon we will be able to give you a response to that.
Okay, one last question from my PhD mentor, Dr. Hong Chen. Beautiful talk.
I don't take questions from her. I'm joking.
Other than reelin, are there any potential lymphoendocrine factor that showed up in your screen?
Can you repeat, Charley? Sorry.
Other than reelin, is there other lymphoendocrine factors that showed up in your screen?
Well, yes. So in the screen, there are, I forgot, there are a lot of them. So the way Charley somehow narrowed initially potential candidates was because she also did RNA-seq of the mutant heart, and to see it was something that was some type of crosstalk. And I think from that, Charley narrowed to potentially 12 or something like that, that may be on the top of the list to start screening. Among those was reelin. And some of those are the ones that are next on the screening approach to see if indeed they do something as well. I think we mentioned in the paper that it's likely that reelin is probably cooperating with some other factors, and maybe lymphoendocrine as well. But again, at the moment, we don't have a conclusive answer for that.
Great, thanks, Guillermo.
You're welcome.
So, okay, right. So the other follow-up question quickly is: the reelin has any impact in the lymphatic endothelial cells itself? Does reelin?
Well, besides what was reported in Tahias Makinen's paper, I don't think Charley has seen any other. Again, we haven't focused, to be honest, in the entire vasculature to see if there are other defects or not. And besides, we recently have enough, because the problem really in the standard rules are kind of difficult to work because of the lethality and the number of defects they have. So we recently started to generate enough conditional, lymphatic specific conditional reelin mutants that will really allow us to more precisely address the kind of questions you're asking, whether they are associated defects or what other things reelin is doing. So the same is true. Hopefully soon we will have a reelin gain of function in our hands that will allow us to also complement those studies in many different ways.
Yeah, beautiful. Thank you.
Thank you so much, Guillermo. Thank you so much, Charley. It's a beautiful work. We're already over time, but if you have a question that you did not get to ask, please email the question at info@navbo.org, and we'll make sure that we share the question with Guillermo.
Oops.
I hope you found this webinar as exciting as we did. These webinars are brought to you by the NAVBO Education Committee. Our next webinar, as Guillermo mentioned, is on Thursday, April 1st, and we'll be hearing from Dr. Karen Hershey on the regulation of endothelial cell specialization. Look for more information on the NAVBO Newsbeat. And please fill out the evaluation form at the end of the webinar or when you receive it in a subsequent email. Let us know what other topics or speakers you may want to see in the future. Thank you so much for joining us today. Thanks again, Guillermo, and everyone have a beautiful day.
Thank you. Have a good one. Bye. Oh, I disconnect from me.